So in this next video, we're going to be talking about how things move in and out of the nucleus. And the nucleus is the equivalent of a cell's corporate headquarters. And its main functions include a variety of things, including maintaining the cell's genetic information, which is in the form of DNA. It also is the place where this DNA is copied in a process called replication, and this occurs before cell division takes place. As we talked about, this is also where ribosomes are made, and they're made in this special location in the nucleus called the nucleolus. And these ribosomes are made up of protein and rRNA. It's also the place where DNA is processed into messenger RNA or mRNA, and this process is termed transcription. And it's also where gene transcription is regulated. So this is regulating when a cell copies their DNA. So in this video, we're going to be talking about transport into and out of the nucleus. And the nucleus is the equivalent of a cell's corporate headquarters. And there's a lot going on in the nucleus. For example, in the nucleus, the cell is maintaining its genetic information, which is in the form of DNA. It's also replicating the DNA or copying the DNA before cell division. It's the place where ribosomes are made, and ribosomes, as we talked about, are made up of protein and ribosomal RNA or rRNA. It's also the place where DNA is processed into messenger RNA or mRNA, and this process is termed transcription. And then there's a lot of things being regulated. So when a cell copies their DNA is regulated, and when a cell transcribes particular genes, and genes are protein encoding sequences. And so there's a lot happening in DNA, and so there has to be some sort of traffic occurring between where proteins are made in the cytosol and where a lot of this action is taking place. So we're just going to talk about a few examples for outbound and inbound traffic. So the first one we're going to talk about is transcription. So mRNA, right, is copied from DNA. So here we have this complementary sequence of mRNA being made where if there's a T, then we have an A or an A and a U. And this is going to be used, this mRNA, as we just talked about, is going to be used by ribosomes to make proteins either in the cytosol or in the ER. But that means that this mRNA actually needs to move out of the nucleus. And so once it moves out of the nucleus and into the cytosol or into the ER, then we can convert the mRNA into protein. And so that's one reason for outbound traffic. This process also, as we talked about, requires ribosomes. But it just so happens that ribosomes are made in the nucleolus, right, of ribosomal protein and ribosomal RNA. And so those also need to move out of the nucleus. And things need to move into the nucleus. So just as examples, if the proteins are made in the cytosol for ribosomes, ribosomal proteins, they need to go into the nucleus to be assembled into ribosomes. We also need to import in materials for DNA replication. These can include deoxyribonucleotides and enzymes that catalyze this process. And we need materials for transcription. So ribonucleotides, transcription factors, and other enzymes that regulate and coordinate this process. So there needs to be a way to get material in and out of the nucleus. Okay, so how does the nucleus regulate its traffic? So we're just going to start by introducing some key structures and regions in the nucleus. So the nucleus is highly organized and it's very large. It's the largest organelle in a cell. And key regions include the nucleolus, that's this electron-dense region here, where the ribosomes are being assembled. It's covered by this double membrane. So there's two lipid bilayers stacked on top of each other. And this is called the nuclear envelope. Just under the nuclear envelope, we have the intermediate filament, which is the nuclear lamina, and this provides structure and rigidity to the nucleus, and so it's not squashed. when the cell is moving, or when there's compression forces. And then studded into the nuclear envelope are these pore-like openings that are around 60 nanometers in size. And so you can see them better in this cartoon here, where we have our double membrane, our nuclear envelope, and then there's holes in that nuclear envelope, which are shown here in green. So those holes are nuclear pore complexes. And here's a very high-powered electron micrograph showing that there's this place. So here's one membrane, and here's another membrane, so that's the nuclear envelope. And there's this place where essentially it looks like it's pinched together. And in this area where the nucleus essentially is bridged are these nuclear pore complexes, and they connect the inside of the nucleus with the cytosol. And these complexes are big. They consist of about 30 different proteins, and they can regulate what goes into the nucleus and what comes out. So how do they do this? Okay, so here's my cartoon schematic of my nuclear envelope. That's a double membrane. And here is a picture of my nuclear pore complex. It's got this basket region, and then these arms kind of sticking up here that give affinity for different proteins. So small molecules can freely diffuse through the nuclear pore complexes. And so small molecules, I mean anything below 60 kilodaltons. So these can be nucleotides or very small proteins. But researchers observed early on that when they injected some proteins into a cell, those proteins never made it into the nucleus, where when they injected other proteins of similar sizes, they were both large proteins, they did make it through. And so this suggested that this large molecule transport is actually selective. And this makes sense. We don't want random things getting into the nucleus if we don't need them to be there. Things like proteases, for example. So what determines whether a large molecule will enter the nucleus? And as you might guess by now, there's a signal. So the nuclear proteins contain a 17 amino acid long nuclear localization signal. And this serves as a zip code that marks them for transport through the nuclear pore. And so the zip code is going to be recognized by a carrier, and then it's going to bring it through the mail slot that's the nuclear pore and allow them to enter into the nucleus. And we're not going to be talking about the carrier for the nucleus. So how do we know, though, about these sequences? What experimental evidence do we have that these nuclear localization signals exist? And so there were these early and great experiments done by this protein called nucleoplasmin. These proteins are really important to maintain DNA's tertiary structure, so when they wrap around histones. And nucleoplasmin has two parts to the protein. It has these core regions that are shown by the balls here, and these tail regions that are these long parts. And in this experiment, researchers hypothesized that the tails of nucleoplasmin will traffic to the nucleus because that's the part of the protein, the tails, that contains the NLS. So this is a good hypothesis because we have our guess and our mechanism. We think that there's going to be an NLS in this tail. And so what they did was they separated the head regions. They were able to separate the head regions from these tail regions. And then they labeled these with fluorescent markers. And so we could follow either where the tails went when they were injected into cells or the heads, okay? And they're thinking that, their prediction, if this is true, if the tails have the NLS, then when this part of the protein, when it was separated into two parts, the tail fragments will localize to the nucleus, and the core, this part of the protein, will remain in the cytosol. And so here were their results. When they injected the tail fragments and waited, sure enough, those tail pieces went to the nucleus. And when they looked at the cells where they had injected the core fragments, those core fragments remained in the cytosol. So this suggested, again, that there was this nuclear localization signal. and then follow-up work characterized this 17 amino acid sequence. It's a conserved sequence that directs proteins to the nucleus. And that's because the NLS is recognized by a carrier protein. And this carrier protein, again, is analogous to a mail carrier that can deliver it through the nuclear pore. And because of this NLS, this sequence, is actually sufficient to carry a normally cytosolic protein into the nucleus. And so that's shown here. Here we have pyruvate kinase. So this is a cytosolic enzyme used in glycolysis. And this pyruvate kinase is tagged to a green fluorescent protein. So we can look at this under a microscope by looking for green. So anywhere there's green, that's pyruvate kinase. And when we attach the amino acid sequence for the nuclear localization signal to pyruvate kinase, and we put that pyruvate kinase into cells, what happens? It goes from this area on the outside straight into the nucleus. And so the NLS is actually sufficient to carry any protein into the nucleus. Okay, and finally, because this reduces entropy, right, we're organizing something, essentially, this process requires energy. So this was a short video, but the take-homes are that nuclear localization signals bring large molecules into the nucleus, and these signals are about a 17 amino acid stretch. And proteins with these NLS include transcription factors and proteins involved in DNA synthesis and replication, as well as nucleotides, as well as other things involved in transcription. Nuclear export signals also bring molecules out of the nucleus. We didn't talk about these, but those molecules include things like ribosomes and mRNA, and this process requires energy. So that's it for this video, and on to the next.